Imagine for a second that you're looking at a satellite image of a city right after a massive earthquake.
Okay.
From way up in orbit, you can see the collapsed buildings, right? You can see the rubble blocking the streets. But what you absolutely cannot see are the underlying fault lines that are continuing to shift.
Right. Or the emergency crews on the ground.
Exactly. You are completely blind to the real-time ground-level action. And believe it or not, that is exactly what it's been like trying to treat multiple sclerosis, or MS, for decades. So, welcome to the Deep Dive. Today, we're exploring a frankly groundbreaking January 2026 study.
Published in Nature Communications.
Right. And our mission today is to unpack a revolutionary new radioactive tracer. It acts almost like a GPS tracker for the specific immune cells that cause MS. We're talking about a technology that lets us see the damage occurring at the cellular level before the structural scars even form.
It really is a paradigm-shifting piece of research, because for the nearly 3 million people worldwide living with MS, this isn't just about taking a slightly sharper picture of the brain and spinal cord.
No, not at all.
It's about changing the entire way we understand and treat neuroinflammation. I mean, for a long time, clinical neurology has had to rely on looking at the aftermath of that inflammation. This new technology allows us to track the active immune cells driving the disease. And crucially, it lets us measure whether a disease-modifying drug is actually working in real time.
Instead of just waiting months to see if a patient gets worse.
Exactly.
Okay, let's unpack this. To understand why this is such a massive leap forward, we really need to understand the cellular battlefield. Most of you following neurology know that MS is fundamentally an autoimmune condition, right? It's where the body attacks its own myelin.
The protective insulation wrapped around the nerves.
Right. But the actual mechanics of which cells are doing the damage is where this paper focuses.
Yeah, when we talk about the immune system, we generally divide it into two main branches, the adaptive and the innate. Historically, the adaptive immune system gets almost all the spotlight in MS research.
Those are the T cells and B cells.
Right. You have pro-inflammatory T helper cells secreting cytokines to amplify the inflammation, and B cells producing autoantibodies. But the true focus of this study, and frankly the unsung driver of both the progression and the potential remission of MS, is the innate immune system. Specifically, a group of cells known as myeloid cells.
Let's break down those myeloid cells.
Yeah.
Because they really are the main characters of our story today. You can think of them in two main categories. First, you have microglia. These are the brain's resident immune cells. I like to think of them as the local neighborhood watch. You know, they live there, they patrol the area, and they keep things tidy. But then you have macrophages.
The infiltrating cells.
Exactly. They circulate in the blood and only cross over into the brain and spinal cord when there's an emergency. Almost like a heavily armed SWAT team called in from out of town.
That's a really great way to visualize it. And according to the pathology data, these activated microglia and the infiltrating macrophages are the predominant cells found in active MS lesions.
They're doing the actual damage.
Yeah, they're the ones physically phagocytosing or eating the myelin and releasing neurotoxic mediators that damage the nerves.
Which brings us right to the imaging problem, and frankly, the human toll of this disease. The current gold standard for diagnosing and staging MS is the structural MRI. But returning to our earthquake analogy, the MRI only shows us the rubble.
Just the aftermath.
Right. It shows the structural lesions, the demyelination that has already happened. It tells you where the fire was, but it completely lacks molecular specificity.
Right. An MRI cannot tell you what those myeloid cells are currently doing. It really struggles to differentiate between an old inactive scar from 10 years ago and a site of raging active inflammation driven by these cells right now.
That has to be a terrifying prospect for a patient.
It is. You might be sitting in a neurologist's office looking at an MRI that is technically stable because no new massive lesions have formed, while at the cellular level, an entirely new flare-up is already brewing.
Now, I know scientists have tried to build molecular trackers before to solve this exact problem. There's a PET scan tracer called TSPO that gets talked about a lot. Why didn't that solve the blind spot?
TSPO PET was a valiant effort, but it has some fatal flaws when it comes to clinical application. It was designed to target a specific protein, the 18-kilodalton translocator protein, but the first issue is that it lacks cellular specificity.
So it binds to everything.
Not everything, but it doesn't just bind to those angry, activated microglia. It also binds to blood vessels, to astrocytes, and to peripheral cells. You get a lot of background noise. But the much bigger problem is genetic.
The polymorphism, right?
Exactly. There is a genetic polymorphism, essentially a mutation in the human TSPO gene. Because of this, the physical shape of that target protein changes slightly from person to person.
So the key doesn't fit the lock the same way for everyone.
Precisely. That means the tracer's ability to bind to the target changes depending on the patient's genetics. You could have two patients with the exact same level of neuroinflammation, but their PET scans would look completely different because of their DNA.
Wow.
You simply cannot use a tool like that as a reliable, universal biomarker.
That sounds like a total nightmare for running clinical trials. You'd never be able to accurately compare scan results across a diverse group of patients. So let's just pause here for our first 5-minute recap to lock in what we've covered.
Sounds good.
Okay, bullet 1. We know that MS is heavily driven by these activated myeloid cells, the resident microglia and the invading macrophages. Bullet 2. Our current tool, the MRI, only shows us the structural damage after the fact. It's the rubble. And bullet 3. Older molecular trackers like TSPO PET are just too noisy and genetically variable to trust.
That sums it up perfectly.
It really puts you in the shoes of the doctors treating this. Let me throw a question to you, the listener.
Yeah.
Have you ever had to fix a problem where you could only see the aftermath, not the cause, like trying to fix a leak in your house, but you're only allowed to look at the water damage on the floor, never the pipes in the ceiling? How much harder does that make finding a solution?
It makes early intervention incredibly difficult.
Yeah.
But that exact problem is what this new tracer was engineered to solve.
Enter the actual innovation in this paper, 18F-fluorimidrimer. And since neither of us wants to stumble over that chemical name for the rest of the deep dive,
Definitely not.
let's stick with the abbreviation the researchers use, which is 18F-FMD.
Scientifically, 18F-FMD is a fluorine-18 labeled synthetic 4th-generation polyamidoamine hydroxyl dendrimer.
Okay, that is a massive mouthful of chemistry jargon.
It is, but the physical structure of it is what makes it so brilliant. Let's break it down. Polyamidoamine just refers to the synthetic polymers it's built from. Hydroxyl means it has water-loving molecular groups on the outside, which helps it stay stable in the body.
And the dendrimer part.
Dendrimer comes from the Greek word for tree. It means it has a highly branched structure.
Okay, let's translate that structure into a visual memory for everyone listening. Picture this dendrimer like a microscopic, highly branched tree. Or maybe a perfectly symmetrical synthetic 3D snowflake. It's built out of these synthetic polymers, engineered at a very specific, tiny size. And attached to the branches of this microscopic tree is a tiny radioactive tag. That's the fluorine-18 part. That radioactive tag is essentially a flare gun. It's what allows the PET scanner to see exactly where this tree travels inside the human body.
What's fascinating here is how this synthetic tree actually behaves biologically. Unlike older tracers like TSPO, 18F-FMD does not try to bind to a specific receptor on the surface of a cell.
Which is a massive paradigm shift.
Right. By ignoring receptors entirely, it completely sidesteps a genetic mutation problem we just talked about. Instead, it takes advantage of the hyperactive behavior of the disease cells themselves.
How so? Like, what are they doing differently?
It relies on a process called fluid phase endocytosis. When myeloid cells, like our neighborhood watch microglia, become activated and angry, their metabolism shifts. They become hyperactive and start constantly gulping the extracellular fluid around them.
Like they're rapidly tasting their environment to look for threats.
Exactly. They are literally chugging the fluid. So this dendrimer acts as a passive Trojan horse. It just floats in the fluid. Resting healthy cells, the ones just peacefully monitoring the tissue, ignore it because they aren't aggressively gulping fluid.
But those angry activated myeloid cells drink it right up.
They do. And because of the dendrimer's specific synthetic structure, once it gets swallowed, it gets trapped inside the cell's lysosomes.
Which act kind of like the cell's stomach, right?
Precisely. It gets trapped and the radioactive signal concentrates right at the site of the angry cells. But for this to happen specifically in the central nervous system, there is a crucial prerequisite, a dysfunctional blood-brain barrier.
Right, because normally nothing gets past that barrier.
In a healthy person, the blood-brain barrier is tightly sealed, and this dendrimer is simply too big to cross it. It just circulates in the blood, filters through the kidneys, and ends up in the bladder.
But in an active MS flare-up, that barrier is compromised.
Exactly. The localized inflammation causes the blood-brain barrier to break down. The dendrimer slips through those specific microscopic cracks, enters the central nervous system, and gets immediately gobbled up by the hyperactive myeloid cells waiting on the other side.
And because it's trapped inside those specific cells, the PET scan lights up with incredible precision exactly where the active inflammation is happening. It's beautifully elegant in its simplicity. Let's pull these threads together for our second quick recap.
Go for it.
Bullet 1. 18FFMD is a radioactive tree-like nanoparticle. Bullet 2. It requires a dysfunctional blood-brain barrier, which naturally happens in MS and to enter the central nervous system. In bullet 3, it is specifically gulped up by activated angry myeloid cells, while resting cells ignore it. So, to you listening, think about it like this. If you were designing a security system, would you rather track the structural damage a burglar leaves behind, like a broken window? Or would you rather put an invisible glowing dye directly on the burglar's tools? That is the kind of upgrade we are talking about here.
It's a massive upgrade conceptually. But of course, a brilliant concept in chemistry doesn't mean much until you prove it actually works in a living, breathing, biological system.
Right. And here is where it gets really interesting, because the researchers designed an incredibly elegant experiment to prove it. They use something called the EAE mouse model. EAE stands for Experimental Autoimmune Encephalomyelitis,
Which is the standard animal model used to simulate MS.
Exactly. For those not deep in the lab science world, they induce the disease using a specific peptide that tricks the mouse's immune system into attacking its own myelin. It perfectly mimics what happens in human MS.
And the clinical progression of the EAE model is highly predictable. That makes it perfect for testing timelines. The physical symptoms ascend rostracaudally.
Meaning the weakness starts at the tail and moves up the spine toward the head.
Right. Generally, by day 12 after induction, you see the mice showing distinct physical symptoms. Tail weakness progressing rapidly into hind limb paralysis.
But the researchers didn't wait until day 12 to scan them. They put these mice in the PE scanner at the presymptomatic stage, specifically on day 8. And this presymptomatic discovery is what really caught my eye.
It's incredible.
Even before these mice showed any physical weakness, the 18FFMD PE scans were lighting up bright red and yellow in their lumbar and cervical thoracic spinal cords. The tracer found the cellular fire days before the disease caused any physical paralysis.
To really understand why that signal was so bright and why it happened so early, we have to look at the flow cytometry data. Flow cytometry is a technique that lets researchers take a tissue sample and literally count and categorize every individual cell.
The numbers here are just wild.
They are. The baseline data versus the day 8 data is staggering. In a healthy, naive mouse, resting peaceful microglia make up about 69.9% of the immune landscape in the spinal cord. It's a very calm environment. But in the EAE mice, by day 8, that number plummeted to 20.28%.
Wait, if the resting microglia dropped from roughly 70% down to 20%, those cells didn't just vanish. They must have transformed.
Exactly. They became the aggressors. The flow cytometry data showed that activated angry microglia spiked from a baseline of just 0.13% to over 20%.
That is a 160-fold increase.
A 160-fold increase in the population of disease-driving cells. And on top of that, the infiltrating macrophages that peripheral SWAT team called in from the blood skyrocketed from 0.88% to 37.53%.
So the spinal cord went from a quiet, balanced environment to a highly inflammatory battlefield, absolutely dominated by these invading macrophages and activated microglia.
It's like takeover.
But I want to play devil's advocate for a second. A skeptic might look at this and say, well, the blood-brain barrier is broken. Maybe this radioactive tree is just passively leaking in and sticking to everything like wet paint. How do they actually know it's only the bad actor cells eating the tracer?
That is a vital question, and the researchers anticipated it. To prove the specificity, they used a very clever validation tool. They created a fluorescent twin of the tracer called C-Sci5HD.
A twin?
Yeah, it has the exact same chemical structure, the exact same molecular weight, and the exact same biological behavior as our radioactive 18F-FMD. But instead of a radioactive tag that you see on a PT scan, it has a fluorescent dye that you can see under a microscope.
So they can actually map it cell by cell.
Precisely. They injected this fluorescent twin into the mice, waited, and then analyzed the spinal cord cells under a high-powered microscope. The results were visually definitive. The fluorescent dye was found almost exclusively inside the infiltrating macrophages and the activated microglia. The uptake in the angry microglia was 9-fold higher than in the resting ones. And the adaptive immune cells, the T cells and B cells, completely ignored it. They didn't eat the tracer at all.
That is a massive finding. Proving that T and B cells ignored it confirms that this tracer is hyper-specific to the innate immune system. Let's do our third recap to lock in what this specific experiment proved. Bullet 1. 18F-FMD detected immune activation in the spinal cords of mice days before physical paralysis even began. Bullet 2. Cellular data proved a massive shift, from resting to activated microglia and invading macrophages. And bullet 3, fluorescent tracking confirmed only the specific bad actor cells absorb the tracer. So, listener, what stands out to you about the ability to detect a disease at the cellular level before any physical symptoms appear? Imagine what that kind of early warning system could do for long-term patient outcomes.
It changes the entire timeline of intervention. But the researchers knew that an early warning system is only half the battle. It's one thing to watch a fire start. It's another to know if the fire hose is actually putting it out.
Exactly. Which brings us to the ultimate so what of this paper. If we can see these cells with this tracer, can we use it to test if an MS drug is actually working?
The short answer is a resounding yes. To demonstrate this, the study tested 2 very different therapies on these EAE mice, starting treatment right at that presymptomatic day 8 point when the scans first lit up.
What was the first drug?
The first drug they used was FTY-720, more commonly known as fingolimod. This is an FDA-approved MS drug. Its mechanism is a bit complex. It's a sphingosine-1 phosphate receptor modulator, but practically speaking, it traps lymphocytes, like T cells, in the lymph nodes.
So it keeps those peripheral troops locked in the barracks and out of the brain.
Exactly, which indirectly calms down the overall central nervous system inflammation.
That's the established drug. But the second therapy they tested was brand new, and frankly, it's a brilliant piece of bioengineering. It's called H74DS3M8. We'll just call it dendranib to keep it simple.
Good.
This drug is actually built on the exact same hydroxyl dendrimer tree scaffold as the tracer. But instead of carrying a radioactive tag to light up a scanner, it's engineered to carry 8 individual payloads of a therapeutic drug called dasatinib. It's like a tiny tree carrying 8 apples made of medicine.
It's a masterclass in targeted delivery. Because dendranib uses that same dendrimer structure, it gets selectively eaten by the exact same hyperactive myeloid cells we've been tracking. It ignores the healthy cells.
Just like the tracer.
Right. And once it's gulped inside the angry microglia, it acts as a highly potent inhibitor of an enzyme called CSF1R. Now, when pharmacologists look at drug potency, they look at a metric called the IC50. That's the concentration needed to inhibit a biological process by half.
Okay.
The pharmacology data here showed it stops microglial proliferation at an incredibly low concentration. It has an IC50 of 343 nanomolar.
Meaning you don't need to flood the patient's entire system with toxic levels of the drug. A tiny targeted amount does the job.
Exactly. And the most elegant part of its mechanism is that it stops the microglia from multiplying without causing cell death. It doesn't kill them. It essentially forces the hyperactive, angry cells to calm down and revert to a resting state.
It's like sending in a highly specific hostage negotiator instead of calling in an airstrike on the whole building. You save the tissue while stopping the riot.
So how did the mice actually fare on these 2 drugs?
The clinical results were night and day compared to the control group. The mice that received a fake vehicle treatment hit severe paralysis scores, averaging about a 2.7 on the clinical scale. But the mice treated with either the established fingolimod or the new dendranib stayed way down around a 0.4 to 0.6.
They showed mild to almost no physical symptoms.
The clinical rescue was fantastic, but the real triumph of the paper, the reason it's in Nature Communications, is what the PT scans showed. The 18F FMD PT scans mirrored that clinical success perfectly.
The scans actually changed dramatically. In the treated mice, those bright, raging warning signals we saw in the spinal cords dropped 5 to 8-fold. The imaging returned almost to the dark, quiet baseline state of the healthy, naive mice.
The tracer dimmed in real time as the drugs work. It proved exactly what it was designed to do. Okay, final recap to synthesize this section. Bullet 1. The tracer successfully monitored the effectiveness of 2 distinct MS treatments. Bullet 2. Fingolimod works by trapping immune cells, while the new dendranib stops microglia from multiplying. And bullet 3. The PET scans dimmed in real time as the drugs worked, proving 18F-FMD's value as a biomarker for clinical trials.
If we connect this to the bigger picture, the translational impact of this paper is immense. This is not just an elegant mouse experiment that's going to sit on a shelf gathering dust. The synthesis of 18F-FMD has already been scaled up and optimized for clinical use.
It's already out of the lab.
It is. In fact, it is currently being evaluated in human patients right now in an ongoing Phase 12 clinical trial number, NCT 05395624.
And that is where the real-world value lies. Treating MS is incredibly difficult because the disease is so heterogeneous. It looks different in every single patient. Historically, a patient would start a new disease-modifying therapy, and then they wait. Their doctor would have to wait 6 months or a year to take another structural MRI just to see if new permanent scars had formed.
Exactly. You were essentially waiting for the brain to suffer irreversible damage just to evaluate if the therapy was ineffective. What 18F-FMD offers is the power to stratify patients accurately and quickly. You give the patient the drug, you do a PT scan shortly after, and you can see immediately at the molecular cellular level if the innate immune activation is calming down.
If the signal dims, you know the drug is working. You stay the course.
And if the signal is still raging, you pivot to a new therapy immediately, before the irreversible demyelination occurs.
It takes the terrifying guesswork out of disease-modifying therapies. We are moving from looking at the rubble to tracking the wrecking crew in real time. Which brings me to one final thought for you to mull over as we wrap up today's deep dive. As we've seen today, this tracer lights up specifically when the innate immune system, these myeloid cells, goes rogue. But innate immune activation isn't just unique to multiple sclerosis. It is fundamentally the driving force behind the progression of Alzheimer's, Parkinson's, and ALS.
That's a very good point.
So could a simple microscopic radioactive tree be the universal key to finally mapping and maybe stopping the hidden immune battles of the human mind?
It's a profound possibility, and it raises an incredibly important question for the entire field of neurology to explore over the next decade.
It absolutely does. Thank you for joining us in this deep dive. Keep questioning, keep learning, and we'll see you next time.